Since the 1990s, the European native ash, called common ash, or in Latin Fraxinus excelsior, has been threatened by a pathogen called Huminuscyphus fraxineus. Most likely the pathogen was introduced from Asia into Europe with living plant material. Hi, my name is Lene Rosgaard Nielsen and I'm Associate Professor at the Department of Geosciences and Natural Resource Management at the University of Copenhagen. During the last many years, I've been interested in ash and ash dieback. It's well known that global travel and trade has increased markedly over the years. And this has also caused an increase in the numbers of introduced pests and microbial pathogens to new areas. In most cases, an exotic pest or pathogen will not be able to survive in a new area where its host is not present. However, sometimes the insect or pathogen will be able to find a suitable host. In worst-case scenarios, the insect or pathogen thrive very well on the new host, which lacks defense mechanisms. And such a host shift can trigger the outbreak of a new emerging infectious disease. The consequences can be severe, with high negative impact on the host and the flora and fauna associated with the host, as is the case with common ash. Common ash is an iconic species. In Norse mythology, tree Yggdrasil, the tree of life, was an ash tree connecting realms by its branches and roots. Museum shops often sell pendants with the tree of life and other items with tree Yggdrasil. Nowadays, the species has great importance for biodiversity, as it is a key species at riverine ecosystems and offers home for many species of insects, fungi and lichens. In Danish forestry, it is common on moist, fertile soils. The wood is treasured, especially for furniture and floors, but it also makes a very nice flexible handle for tools. In terms of volume, it plays no or only a little role in wood industry in the Nordic countries, but it is nevertheless a valued species by silviculturists in the southernmost part of Scandinavia. We are now at Tusenæs, at a former clonal seed orchard of the Nature Agency, and we can see the symptoms on all the trees. Here we see a heavily infected tree. We can see that the twigs are discolored and the branches become girdles and then they die. And when we look into the crown of such a tree, you can see that the primary crown is more or less dead. And then there are a lot of these shoots here, epicomic shoots, found all over the crown. So if we cut down such a diseased tree and look into the trunk, we will see discoloration of the wood. So how does the trees become infected? Well, the pathogen produces a lot of fruit bodies, also called ascocarps. And in these fruit bodies, there are a lot of spores, ascospores. And these are then spread by the wind, and then they land on the leaves of the trees and infect the leaf. And then in the case of common ash, it doesn't stay in the leaf, but it enters into the twigs and into the tree. And then we see all these symptoms in the crown. In the fall, the leaves will fall off, land on the forest floor, and here the pathogen then acts as a decomposer of the leaves. The following year, again in July, August, new fruit bodies will develop and we will get a production of new ascospores that again will be spread by the wind, land on the leaves, and we will have a new infection. We don't know exactly when the pathogen arrived in Europe. The first symptoms were observed in Poland in the 1990s, and then in the Nordic region in Sweden, it was observed in 2001 for the first time. In Denmark, we saw symptoms in 2002. And then in the southern part of Norway, it was first observed in 2006. Then in the southern part of Finland in 2007. And now approximately 25 years after the pathogen was first observed, it's found almost in the entire distribution range of common ash in Europe. Luckily, there is a small fraction of trees that tolerate the pathogen much better than others. And that is very clear here in a place like this as Tuse Næs. This place was originally established with the intention of seed production, and 39 trees were selected from different places in Denmark. These were then grafted 25 times each and planted out here in Tuse Næs. When the pathogen came, we did not have 25 copies of each genotype any longer. Many of the trees died, and only few seemed to stay healthy. This tree here, tree 35, is the healthiest tree here in the trial. We have assessed the crown damage of these trees since 2007, and we see a very clear signal that there is strong genetic variation among trees in how well they can manage the pathogen. And this is very similar to findings of our colleagues from other places in Europe. We have also looked at progeny from other trials in Denmark, and we see that there is a clear signal. If you are a healthy tree, there's also a higher probability of having healthy offspring. So the trait seems to be inherited from parents to offspring. And this is, of course, very good news in relation to selection, natural selection as well as artificial selection. Now, one thing is the situation in the clonal seed orchards and the young plantations. Another thing is what's going on in an old forest stand, as the one that we're visiting here today. This was a question that my colleague, Erik Dahl Kjær, he had a PhD student that wanted to look into these things. So Devrim Semisa-Kooming, she went here to Valby Hegn in northern Zealand, and here she collected tissue from small seedlings of ash, and she genotyped the seedlings with microsatellite markers. She also genotyped the mature trees in the forest stand, and then she could assign the seedlings back to the parents and see which ones were mothers and fathers for the seedlings. Then she also looked at the health of all the mature trees, and her results were really clear. The healthy trees contributed much more to the offspring than the unhealthy trees. And that is, of course, good news, because that could then look like that there is already selection going on in the favor of the more healthy trees. Another thing that she saw from her results were that trees from the forest edge, they spread their pollen and seeds quite far into the forest stand. So it seems that it's actually possible to assist the development by planting healthy ashes at the forest edge and then let them spread their pollen and seeds into the forest stand, and then they can outnumber the unhealthy trees in contribution to the next generation of ash. We are now at the Arboretum in Hørsholm, where a lot of our research on ash and ash dieback has been taking place over the years. We still have many ongoing activities here. The last few days we have been transplanting a lot of small ash seedlings. This is some work that is related to breeding, and it's led by my colleague Erik Dahl Kjær. And we will now go and meet Erik here in the Arboretum. So Erik, we now know that there's genetic variation among ash trees in ash dieback resistance and that the trait is inherited. So don't you think this is the perfect setup for breeding activities?
Yes, indeed so. And actually all the way back in 2008, when we first realized that we had this heritable variation, we discussed with the Nature Agency and also the private forest association how to utilize that to develop more resistant trees for Danish forest. And there's been a lot of activity since then. The foresters have been selecting healthy trees in the forest that survived the disease and being grafted into seed orchards. And today there are established 4 clone seed orchards. And actually we have there's also a 5th seed orchard which was just established just before disease came, that was converted into one of these so-called healthy seed orchards. So a lot of things have happened, a lot of activities, been a huge effort, and as scientists we've been part of that. And it's actually an ongoing effort. As you can see here, we are now in this unique situation that we can actually establish a second generation seed orchard, only 12 years after we started. So based on the 2,400 trees that we followed since 2006, we really followed them year by year, and the few trees that really managed well during the infection has been selected. And the female ones, as you know, of course, there are male and female ash trees. So from the female ones we could collect seed and sow the seed in these boxes here. We did that last spring. And they've been standing here in the arboretum and been subject to infection from the natural infection. So it's kind of mimic the forest floor. And most of them look healthy, fortunately, but we can actually see that when we go through them, there are some of the progenies here that actually have clear signs of infection. Yeah. I'm sure you'll agree with that. Yes. And then that means that we remove them, and only the healthy ones have been transplanted into our nursery down here. We'll follow them this summer. And in the autumn, the best surviving ones and healthy ones will be transplanted into a second generation seed orchard, where they'll grow for 10 years or so, and where we can follow them. And then hopefully by then, when they start to flower, we have an even better, even more resistant seed source for the forests. But they actually just planted over here.
Okay, should we go and see them?
Yeah, let's do it.
Yeah.
So that's what it looks like. So here we have now all the seedlings transplanted that used to be in the boxes, and now each box has been planted here as a progeny, and we follow them here. We follow them here during the summer. We planted approximately 5,000, a bit more than 5,000 seedlings, and out of these 5,000 seedlings, I expect that we will select approximately the 2,500 most healthy in the autumn and plant them in the second generation seed orchard, which will also be a trial, because we're going to follow them there for the next 10 years, that's the plan at least, and see how it develops. And then after 10 years, most likely 250 or so will be left for seed collection. So for the next generation, the second generation seed orchard. And this is of course a case of really strong selection. In first generation, approximately 7-8% most healthy trees were left for crossing. And then here in the second generation, I suppose actually only 1 or 2% of the most resistant trees will be left for seed collection. This is a case of very strong selection, and that's of course we hope that we really can improve the health of the ash forest by doing this activity.
But don't you think such a strong selection can actually be at a cost of the genetic variation in ash and maybe also some other important traits?
Yeah, that's a really important concern. And that's the reason why we've designed, so the whole selection procedure has been designed in order to make sure that we maintain diversity in the next generation, and also we avoid to change traits we don't want to change. It's actually a part of a Nordic cooperation, this design of second generation seed orchards. So similar activities is going on in Norway and in Sweden. It's a project funded by the SNS Forest Research. And the way we're going to the selection, we will be able. The whole plan would be that we can maintain this diversity. But of course, you know, there's many things we don't know. So, in order to be able to really optimize our selection strategies, we need to have a better understanding and the host pathogen interaction. But in order to do that, we need to go to the university to see this kind of research.
Okay, let's go then. We have taken different approaches in order to try to understand at least some of the mechanisms behind ash dieback resistance. We are now at Department of Geosciences and Natural Resource Management, where we'll meet two scientists, Chatai Kosawang and James Noonan, that both are working on different projects related to ash dieback resistance. So let's go and see them. In the greenhouse, we have all our plant material that is being used for a lot of different experiments. Here we are aiming at identifying genes that are being expressed in resistant ash species and resistant genotypes of common ash, but not in susceptible ones.
It is in a joint collaboration between Norway and Denmark. So we have inoculated different ash genotypes. For example, this is the resistant genotype, and this one is a susceptible one with the ash dieback fungus. And we are going to look at the response after inoculation. So right now we are in the process of looking at differentially expressed genes, and we hope to find genes that can give us biological insights into ash dieback resistance.
Together with European partners, we are trying to identify single nucleotide polymorphisms that are associated to ash dieback, and also other SNPs that are related to other fitness-related characters. And this is something my colleague James is working on.
Hi Lena, yeah that's right. So we're going to identify SNPs within hundreds of ash trees like this one. And we're going to find gene variants among these SNPs to find one generation after the outbreak of ash dieback which trees are most resistant. So these gene variants will be used to give results to policymakers and give insights into future conservation and management practices of our ash stands.
Because of the presence of genetic variation among ash genotypes in ash dieback tolerance, and because the trait is inherited, there are possibilities for selection and adaptation to work in the favor of more tolerant ash genotypes. We can assist the processes, for instance by selecting and breeding for more robust ash. In addition, a better understanding about the genetic mechanisms behind ash dieback tolerance will give us more knowledge that can be used to manage and conserve the species in the future.